Okay, let's first take a look at regulation by certain kinds of ions. The major ionic regulators of proteins are going to be the calcium ions. Now, as we're going to see later, cells keep calcium ion levels very, very low. 1,000 or so less. We're talking fractions of a micromolar concentration. 1,000-fold less than what you find in body fluids, fresh water, salt water, whatever the case is. And the reason for that is that calcium regulates many kinds of different proteins. If you keep the calcium levels high, it activates a whole battery of proteins, and you don't always want to turn these guys on at all times. So cells keep their calcium levels very low inside, and then what happens is they are going to then, when you want to regulate those proteins, you selectively let calcium into the cell, and it activates those proteins and so on. But normally we keep calcium levels low so we don't turn these proteins on. Okay, well, in many cases, calcium can directly bind certain proteins and regulate them. So we could say calcium ions directly binding proteins, direct binding. And there's a number of examples of proteins that are directly regulated by calcium ions. This includes certain kinds of enzymes that require calcium ions for their activity, especially a whole family of proteases, protein digesting enzymes. Many of them are activated directly by binding calcium. Without calcium around, they sit there staring at you. Add calcium in there, and they turn serious predator. Okay? Some proteins in the cell's internal framework, or cytoskeleton, are also directly activated by calcium. Cytoskeleton. Okay. But there's another way we can use calcium regulation of proteins. We can regulate a protein by allowing calcium to bind to a second regulatory protein, activating it, and then that protein goes and binds and regulates a whole battery of other proteins. We could say this is indirect regulation. And the major one in this, in calcium regulate things, is a small protein called Calmodulin. Literally means calcium modulator, calcium regulator. Calmodulin is a little small protein that has a big impact. And it's an interesting kind of protein, because calmodulin has two radically different structural forms depending on whether calcium ions are bound or not. When there's no calcium ions around, calmodulin is inactive. It doesn't do anything. And if you look at the tertiary structure of calmodulin, you see something that looks like a molecular barbell. You have an alpha helix, a long kind of rod of alpha helix. That's a spiral coil of secondary structure. And then at either end, you have a folded up portion, kind of a lobe here and lobe here. And each one of these lobes can bind two calcium ions. Okay, so you bind two calcium ions on each of these lobes. Now when that happens, calmodulin is activated. And what happens to the tertiary structure is so dramatic that if you looked at the two states, you wouldn't even think they're the same protein. It's radically different. Somehow, calcium ions causes this whole rigid alpha helix, this bar on the barbell, to collapse into a floppy chain of amino acids. In other words, no secondary structure. And that means you have the two lobes with our calcium ions connected by a floppy, loose, flexible chain of amino acids. And now the active version is capable of binding and regulating a whole bunch of target proteins. And because that kind of loop between the two lobes is so flexible, that means it can hug a whole slew of different kinds of proteins. So many times you let calcium into the cell, you activate calmodulin. Calmodulin regulates scores of different types of proteins. Enzymes, cytoskeleton, things that activate or shut off genes, transcription factors, membrane transport systems, all kinds of stuff can be regulated by calmodulin. So, calcium ion regulation is clearly very important in cells. Now, an interesting thing, anybody taking physiology or remembering physiology? You may remember that muscle contraction depends on calcium ions getting into the muscle cells. Without calcium ions, muscles will not contract. And the reason why is there's this complex of proteins called troponin, and that set of proteins keeps the muscles from contracting, but when calcium comes in, it allows the muscles to contract. One of the three parts of troponin is a molecule called troponin C. And it turns out, once they got the amino acid sequence, the primary structure, and the tertiary structure of troponin C, what troponin C is basically a muscle-specific version of calmodulin. And like calmodulin, it undergoes a radical structural change when calcium binds, and that allows the machinery of the muscle to bind and start generating movement and contraction. Sorry. So if you take animal systems phys or something like that, and you hear about troponin C, it's basically a fancy specific version of calmodulin. Okay, that covers regulation by binding of ions. Question on that? Anyone have any questions, folks?
Can you spell that T word?
Oh, okay, Troponin C. Troponin C. And we take physiology, muscle physiology, run into that because that's our calcium switch, and it's basically a version of calmodulin. Okay, continuing on with non-covalent regulation, let's take a look at a few examples of regulation by small molecules. And we'll give some examples that we've already seen before to illustrate this. Many different kinds of small molecules can regulate many different kinds of proteins. We mentioned some of those common. Certain types of lipids regulate various proteins. Modified amino acids or straight amino acids in some cases can regulate other proteins. Nucleotides and modified nucleotides can still regulate additional proteins. And a whole host of other types of small molecules can regulate various proteins. Once again, this is non-covalent binding here. Well, let's give a couple examples. We've already run into these. How about the steroid hormone receptors? They're regulated by steroid hormones, which of course are a form of lipid. Now you may recall, steroid hormone receptors, in the absence of the steroid, are just beneath the cell membrane attached to an inhibitor protein. And they sit there staring at you. Add the appropriate steroid, it binds the steroid receptor, causes a tertiary structure alteration. That alteration pushes the inhibitor protein off. It can no longer bind the receptor. And not only that, it causes and exposes what we call a nuclear import sequence, a stretch of amino acids that acts like a ticket to the nucleus. And if that's exposed, that steroid receptor now goes, with the steroid attached of course, now goes into the nucleus, and then it binds to other gene-regulating proteins or transcription factors, and it regulates whole batteries of genes. But here we have an example of a protein, the steroid receptors, that are regulated by steroid lipids. And they do dramatic things. They're in the cytoplasm without the steroid. With the steroid, they go into the nucleus and they regulate many different kinds of genes. So that's one example. Another good example that we briefly covered, G protein. We're going to cover this a lot more when we look at cell signaling. The whole diverse family known as G proteins. And G proteins are regulated by binding GTP, guanosine triphosphate, or by binding GDP, guanosine diphosphate. And G proteins can hydrolyze GTP into GDP. So, they're regulated by modified nucleotide. And when they have GDP bound, they're in one structural state, usually inactive. When they have GTP bound, they're in a different structural state. And what that means is most of the time they're active and then they go off to regulate, bind, and regulate other proteins. So in many, but not all cases, G proteins are active only when GTP is bound, and they're dead in the water when GDP is bound. That's not always the case, but it frequently is. That's another example we got. Now, a third example.